Hello everyone. In this video, we will study about one of the important topics under virology, that is rabies virus. So it can come as a 10 marks question or even 5 marks question for your exam. So it is very important topic. So this rabies virus causes a rapidly progressive and fatal inflammation of the brain and spinal cord in humans and animals. So it is a highly dangerous virus. So death is almost certain in the patients infected with rabies virus. So recovery and survival rates are extremely rare. So rabies virus infection, it is one of the examples for zoonotic disease. Zoonotic disease is nothing but the disease which is transmitting from one infected animal to another animal or to human beings. So I think all of you will be knowing the animal which is mainly transmitting rabies infection, right? So yes, it is dog. So let us move into the morphology and pathogenesis of rabies virus. So rabies virus belongs to the genus of Lissa virus and family of Rhabdoviridae. So when we observe the specimen under electron microscope, we can see the characteristic bullet shape of rabies virus. So remember this key point, rabies virus has a characteristic special shape, that is bullet shape, which measures about 75 nanometer in width and 180 nanometer in length. And we all know there are three important parts in the virus, right? Outermost layer will be envelope, middle part will be capsid, and innermost part will be nucleic acid. So similar way, rabies virus has outer envelope. It is an example for enveloped virus. So outer envelope of the rabies virus is embedded with 10 nanometer long peplomers or spikes, that is glycoprotein G. So you can see in the picture the glycoprotein G spikes. So main function of this glycoprotein G is to attach to the host cell after the entry of virus. So that is when infected animal bites the skin of the person, virus enters the skin. With the help of this glycoprotein G, it attaches to the host cell and then it will invade the body. Then the envelope is internally lined by a matrix protein, and inside the envelope there will be nucleocapsid. So here the symmetry of nucleocapsid is helical. So it is helically coiled and it comprises of single stranded negative sense RNA, nucleoprotein and polymerase protein. So rabies virus is a RNA virus. And then major antigens of rabies are glycoprotein G and nucleoprotein. Next, moving on to the pathogenesis of rabies virus. So source of infection. First one is bite. So we all know that dog is the main source of infection. In 99% of cases of rabies, dog is the main source. And other animals that can also transmit this rabies infection involves bats, foxes, raccoons, skunks, jackals, mongooses and other wild carnivore hosts. And other source of this rabies virus infection involves some of the non-bite exposures like licking by the infected animals or through aerosols or transplantation of the organs from the infected from the infected patient to healthy individual. So it can also transmit the disease. So these are the sequential steps of the pathogenesis of rabies virus infection. Initially, soon after the bite from the infected animal to the human, virus multiplies locally at the site of bite. Then it enters into the peripheral neurons. It spreads throughout the neurons. Then it enters into the central nervous system and infects the central nervous system. Then followed by centrifugal spread to various organs, and finally it will be shedding in the saliva of the infected individual. So when a rabid dog bites to the human. So usually the dogs will be biting to the legs, right? So in the skeletal muscle, the initial virus replication will be taking place. Then the virus binds to nicotinic acetylcholine receptor at the neuromuscular junction, and it invades the axons in the peripheral nerves through retrograde fast axonal transport, and multiplication of this virus takes place in the motor neurons of the spinal cord and local dorsal root ganglia, and it will be rapidly moving to the brain. That's why it is called as a rapidly progressive disease. So as it moves to the brain, there will be infection of the brain neurons will be taking place, and there will be dysfunction of the neurons. After that, there will be centrifugal spread along the nerves to salivary glands, skin, cornea and other organs, and the virus will be shedding through the saliva of the infected individual. So this is about pathogenesis of rabies virus. So in the next video, we will continue with the clinical manifestation, laboratory diagnosis and prophylaxis of rabies virus infection. Thank you for watching.